Hello, everybody. Today we have the pleasure to interview Dr. Nathanial Anderson from the Wellcome Sanger Institute in Kingston in the UK to talk about the recent paper he published on Nature Medicine entitled Transcriptional Signatures Associated with Persisting CD19 positive CAR-T cells in children with leukemia. So first of all, thank you for accepting our invitation. Nathanial, how are you today?
Yeah, hi Carmelo, hi everyone. I'm doing very well. Thanks for asking. I wanted to thank you and the rest of the EBMT for this invitation and the opportunity to discuss my work.
It's absolutely our pleasure. And let's start, if you agree. So may I ask you to give us a very brief overview on CAR-T cells in children and their use, especially in CD19 positive hematological malignancies?
Yep, sure. So I can give you a very broad introduction for those who are new to the field. So historically, the treatment of hematological malignancies, such as leukemia, lymphomas, myelomas, they've included chemotherapy, radiotherapy, and hematopoietic stem cell transplantation. However, with recent advances in tumor immunology and immune-targeted therapies, there's a new avenue of treatment that's presented itself in the form of chimeric antigen receptors, or CAR-T cell therapies. And so these are synthetic T cells that are genetically engineered in vitro to recognize specific cell surface antigens on tumor cells without antigen processing or presentation. So this process includes taking a patient's own T cells from peripheral blood, reprogramming them in vitro to express the CAR, or the chimeric antigen receptor, and then reinfusing them back into the patient. And so these CAR-T cells can now specifically recognize target antigens and rapidly proliferate to exert their anti-tumor effects. So you can imagine this is a great example of personalized medicine as it empowers the patient's own immune system to attack those tumor cells. So currently, B cell surface receptors such as CD19 and BCMA are the most common targets for CAR-T cell therapy. So CD19 is relevant for our study, so I'll focus on that. It's widely expressed during all phases of B cell development until their terminal differentiation into plasma cells, but it's not expressed in hematopoietic stem cells. And so it's important for our paper because we focus on CAR-T cells that are designed to target CD19 in children with B lineage acute lymphoblastic leukemia, or B-ALL. And this is the most common subtype and the most common childhood cancer. And so to reiterate, these are blood cancers that are derived from the B lineage, and they only express on cancer cells. And so they're a very attractive target for these types of CAR-T cell therapies.
Thank you very much. And of course, like sometimes you can see also some specific off-target effect, just like to add on your little overview, because of course the CD19 is also expressed in normal B lymphocytes. So, and this is the reason of the B cell aplasia, right? So. Right. So in this case, we can also have some off-target effect. Right. So this is a translational study based on the CARPALL trial, right? Which uses a particular CAR-T, right? CAR-T product. Yeah. Can you tell us a little bit more about these and the characteristics of the product infused?
Yeah, you're right. So this is a very translational study. It's a great example of cutting-edge genomics and basic science being used to gain insight into like a very real clinical problem. So in order to understand the novelty of our product is we first want to discuss the components of a CAR. So the CAR structure consists of an extracellular antigen recognition domain, a transmembrane domain, and an intracellular signaling domain. And the important part is the extracellular domain that actually recognizes the tumor surface antigen. So previously, one of the lead investigators of our study, Dr. Sarah Garashian, she developed a low affinity single chain variable fragment targeting CD19, and that the CAR was called CAT, like the house pet. And I believe the licensed product is now called Auto1, but for our paper, we decided to keep it as CAT. And so by low affinity, we mean that compared to the currently approved CAR product called Tisagen or Kymriah, the CAT CAR has an over 40-fold lower affinity for CD19. So we're talking about protein-to-protein binding affinities. And so Sarah and the team at the UCL GOSH, so in London, they observed that in preclinical models that the CAT CAR-T cells were more cytotoxic and more proliferative than the currently approved CD19 CAR-T cells. So the team then went on to carry out a non-randomized open-label phase 1 clinical trial treating children and young adults under the age of 24 who were diagnosed with high-risk and relapsed CD19 hematological malignancies, but they were actually all B-ALLs. So they were treated with CD19 CAR-T cells. So 14 of those patients received lymphodepletion followed by an infusion with the CAR-T cells. And at the time of their publication, 13 of 14 actually had those prolonged B-cell aplasias that you were discussing before, which is consistent with the persistence of CAR-T cells. So in our study, we went on to include one additional patient. So the clinical trial cohort was 15, 13 of which had remissions, but 6 of them ultimately did relapse. And so for our study, we had 7 patients that were able to achieve long-term remissions. And so the novelty of our product was the low affinity binding. And so it's different from what is the CAR product that exists currently on the market.
Okay, so now guide us through what was your study design and your hypothesis. You already mentioned you focus on those children who have persisting CAR-T cells and long and durable remissions, right?
Right. So, so limited persistence remains a significant hindrance to the development of effective CAR-T cell therapies, and this is a central theme throughout our paper. So CAR-T cell persistence refers to how long CAR-T cells can stay in the body and in circulation. So the idea is that therapy is a one-time treatment. The aim is to have it in for circulation for months or even better for years. However, some patients, the T cells go away very quickly, and this poses a threat to the patient as there's a higher chance of tumor relapse. And so with this in mind, the aim of our project was to study long-lived or persisting CAR-T cells from the CARPALL trial to better understand the biological determinants of CAR-T cell fitness. So there really wasn't a hypothesis in going into this per se, but we wanted to more understand and establish what persisting CAR-T cells look like, and we wanted to address the mechanisms of how these CAR-T cells could persist over many months or over many years. And so the design was simply to collect, sequence, and characterize long-lived CAR-T cells with the necessary biological controls. And so during the trial, Sarah and her team collected blood and bone marrow from children participating in the study, and they cryopreserved these specimens over a time course of 5 years. And upon completion of the trial, Sarah and her team then teamed up with my supervisor, Dr. Sam Vajati. And so at UCL in Goshen, London, they would perform flow cytometry to sort the cells into normal T cells, CAR-T cells, and everything else. And then she would ship the isolated cells to the Sanger, where we would perform single-cell gene expression and T-cell receptor or TCR sequencing on as many samples as we could. At which point I performed the bioinformatic analysis. So in total, we were able to perform detailed phenotyping by flow cytometry on 11 patients. And then for 10 children, there were sufficient CAR-T cells that we could actually look at the single-cell gene expression. And so there were 73 patient samples, 89 gene expression TCRs. So it can get a little confusing because we have a patient that can give us several time points.
Right.
Each time point, there's blood and bone marrow, and then each blood and bone marrow can give you flow cytometry, gene expression, and TCR. And so it can get very, very convoluted about all the different types and modalities that we looked at. But most of our hypothesis generating data was derived from 89 samples of the single cell gene expression, which is like where the majority of my work was.
Thank you. And now guide us through your first findings. What did you notice first in these samples?
Yeah, so the first thing that I did was quality control to make sure that we're looking at high fidelity, good quality single cell data. And so I threw all the cells from all the patients into one big object, and then I clustered them. So I'd labeled the cells from their patient of origin, their source, so whether they're from blood or from bone marrow, as well as their time points. So the products, which are the infusion products, those are time point 0, so that they go first. Then the earlier time points are months 1 to 3, then mid time points were months 4 to 6, then everything from month 7 all the way to year 5, these would be considered late time points. And that's where the persisting or long-lived CAR-T cells, that's where they come from. So overall, we had four time point bins that we had. What was immediately apparent was that all the CAR-T cells across the patients, they segregated by their time points. So CAR-T cells were transcriptionally distinct and were expressing their own unique set of genes depending on whether they were products, early, mid, or late time points. And then I did the first step, which is the first step in most single cell studies, which is to do cell typing. So what kinds of cells are we looking at? Are they CD4 T cells? Are they regulatory or cytotoxic CD8 T cells? And so to answer that, we just looked at the expression of these lymphocyte markers. And while they did express CD3, they did not express markers like CD4 or CD8. And originally I'd assumed that these were like a technical issue with 10X because of like some kind of dropout. I was like, why are these lymphocyte markers missing? So in terms of the validation, this is when we reached out to Sarah, and she was able to validate what we saw at the gene expression level at the protein level using flow cytometry. So her and her team were able to confirm that the persisting or late CAR T cells actually did not express CD4, CD8, but they harbored a double negative T cell phenotype, which we didn't expect. Their team also confirmed that these weren't gamma delta T cells, and they harbored an effector memory phenotype, which was consistent with our single cell work. And so our first finding was that the CAR T cells look different across time, and that the late time points, they didn't express CD4 or CD8, they appear to have a double negative phenotype, and they also have an effector memory phenotype as well.
Thank you. And how did you validate this?
So the original discovery in the gene expression, we validated at the protein level using the flow cytometry.
With the flow cytometry, okay, good. Yeah. And instead, did you notice differences with regards to transcriptional signatures in CAR-T cells based on their persistence?
Yes, so we had, due to the nature of the clustering, we had an inclination that there were differences in gene expression that we saw at the different time points. And what we appreciated was that these differences would delineate distinct transcriptional signatures. And so the cells of the infusion product, for example, so time point 0, they were enriched for genes related to cell cycle, nucleosome assembly, and glycolysis. And then they also expressed gene modules related to naive lymphocytes and early memory differentiation. But when we looked at the CAR T cells post-infusion, so like from the earlier time points, months 1 to 3, all the way to the late time points at like year 5, for example, these existed on a spectrum of granzyme expression, with earlier populations expressing granzyme H and B, and the late CAR T cells shifting in phenotype towards a granzyme K phenotype. And so we could use the genes that are defining the late or persistent CAR T cells to define a transcriptional signature that's unique to these cells. And so the most recurrent and strongest markers of these generated our persistent CAR T cell signature, which included some well-known immune-related genes like TIGIT or GPR183, but there were other genes with unknown or emerging roles in immune biology. And so the strongest markers of this transcriptional signature included FXYD2, HMOX1, and DEN-D2, for example. And these were strongest at the late time points and were the main driver genes that were separating these T cells from everything else. And because we saw some genes like TIGIT, for example, that we know is an established marker for exhaustion, we then explicitly went to look for co-expression of other exhaustion markers by single cell gene expression and at the protein level by flow cytometry. So we found that the late CAR T cells expressed canonical co-inhibitory receptors such as TIM3 and LAG3, but they didn't express PD-1, for example. And so together with the granzyme K expression, our persistent CAR T cells bore some similarities to a recently described cell type called precursor exhausted T cells. And so the transcriptional status of our CAR T cells suggests that they are still functionally active, proliferative, but they also bear some hallmarks of a near exhaustion-like phenotype that looks very different from the CAR T cells we see at an earlier time. And so the most surprising thing was that even though we broke down the persistent CAR T cells by patient, so before I'm looking at everything together, but if I look at all the late CAR T cells individually in every single patient, all the cells would converge on the same transcriptional signature. So it's highly robust and highly consistent across patients. So I like to think of it like this, that if you achieve a molecular remission, you can already predict what the CAR T cells look like because it's so consistent across all the patients that are able to achieve a molecular remission. And so these will express granzyme K, and they'll also have this very distinct transcriptional signature that's defined by FXYD2, HMOX1, and let's say TIGIT, and they have this precursor exhausted-like phenotype.
I see. And then you interrogated the TCR sequences, which is, from what I gather, more your field, no? Since you're like a bioinformatic together with like the single-cell RNA-seq, right? And you were able to sequence different samples across also different time points. So what did you find?
Right, yeah, it's definitely true. I'm more of a bioinformatics analysis, so I've been learning a lot about immunology and CAR T cells from the hematologist that I work with. So when we did the TCR sequencing, we had defined the immunophenotype, we defined the transcriptional status, and now we wanted to understand where these cells were coming from. So to answer this question, we used the TCR sequencing. So the TCRs are highly diverse heterodimers consisting of a combination of alpha and the beta chain, and they're expressed in the majority of T cells. and they also have the variable region that's encoded by the V and the J genes. And so in a way, these combinations can create almost like a unique molecular barcode, such that we can track clonal populations over time. And so before we were looking at gene expression changes over time, but now we're looking at the identity of the cell, or the clonotypes, and how those change over time. And so originally when I performed this analysis, I had hypothesized that there was an early clone that could expand and that would dominate the clonal landscape of persisting CAR-T cells. Therefore, we'd see that there'd be very clonal, composed of a single or very few clones. After all of these cells appear to be functionally converging on the same transcriptional signature, but that's actually not what we saw. When we sequenced the TCRs from blood draws from the same patient over time, there were different clones detected in each draw. And it was actually very rare to see, to be able to track the clones across different time points. And so what we found was that the vast majority of cells across time points harbored unique clonotypes that were not observed at subsequent time points. And so we concluded that persisting CAR-T cells were therefore quite diverse and polyclonal, and it's not the expansion of one or a few clones that explains the converged transcriptional signature that we're seeing. And it would indicate that there's a functional convergence across different clonotypes onto this transcriptional signature that we're seeing. And it's independent of the different clonotypes that exist.
And so you explained that you found a convergency, right? Like a sort of like a signature that you could identify. Do you think this is private to CAR-T cells, this kind of behavior of T cells? Or, and if so, do you think that also other CAR-T cell products may have this sort of functional convergency? How did you approach this research question?
Yeah, no, it's an excellent question. It's the exact same one that we had when we did this. We wanted to know how prevalent that signature was across different CAR products, because we saw it was so distinct and consistent across our patients. But we were hindered by the fact that there weren't and there aren't very many studies that have probed persisting CAR-T cells with single-cell resolution in human primary data. The one exception to that is a paper that came out last year by the University of Pennsylvania, where they had done a single-cell transcriptomic analysis of persisting CD19 CAR-T cells, but from 2 adults with chronic lymphocytic leukemia. So we're looking at a different age group and a different cancer type, but they were CD19 CAR-T cells. And I would just want to highlight the beauty of open access and collaborative science, because just sending them an email, they're able to send us their data, and I could integrate it with mine very quickly and very easily. And so it was really lovely. And so remarkably, we found the UPenn's persisting CAR-T cells harbored nearly the exact same transcriptional signature as the persisting signatures that we saw in children with B-ALL. And there were some discrepancies between their immunophenotype. For example, they described CD4 positive cells, whereas we had double negative T cells that I discussed earlier. But overall, the transcriptional signature was very similar between these two, and it was undeniable. So together, this data might suggest that independent of the initial CAR product, so we had the CAR-T cells, they have a CAR product like Kymriah, Tisagen. The patient age, we were looking at children, young adults, they were looking at patients in their 50s and 60s. Initial cancer diagnosis, we were looking at B-ALL, they were looking at CLL. It appears that it doesn't actually matter. As long as you have persisting CAR-T cells, they may harbor the same, maybe universal transcriptional signature of persisting CAR-T cells. And this is almost a biomarker of achieving a molecular remission. So we then questioned whether this was private to persisting CAR-T cells. Like, could we see this in a normal physiological context, for example, in peripheral blood, in bone marrow, and thymus, or maybe in tumor infiltrating T cells? Like, where else could we potentially see this signature? And our search came up short. So even in tumors where, with responses to immune checkpoint inhibitions, which are quite immunogenic, we were never able to find the signature outside of the context of persisting CAR-T cells. An important caveat to that statement is that the data sets that we investigated, they were not on a similar scale to ours. For example, some other CAR-T cell studies, when they looked at single cell of CAR-T cells, they looked at the products and they looked from months 1 to 3. And on our scale, that's very early, because we're looking at 5 years infusion. Yeah. And so it could be said that if you just waited a bit and like actually single cell sequence some things months, 6 months, 7 months plus, you might be able to see the signature. But that's highly reliant on the CAR product being able to induce persistence in T cells. And one could also think that if there's like a chronic infection and you actually sequence it years out, you might still see the signature. It could be a signature of like repeated antigen exposure and some kind of chronic infection or chronic antigen stimulation, but no one's actually looked that far out to be able to say so. So there's a bit of an asterisk on our study that we found what we found, but we haven't actually looked in every single context. So it could be more universal than we think.
So you already responded to my next question, which was what's next, right?
Yeah.
So you already have an agenda for your research portfolio, right?
Yeah, exactly. So like the main take home from us is that we've potentially discovered this universal transcriptional signature of persisting CAR-T cells, the implications of which like we're now unraveling, as you mentioned. And so I foresee the next steps being validating the signature, determining how prevalent it is across CAR-T cells. So far, we've only looked at the UPenn group, which had 2 patients. We had less than 10 patients that were able to actually achieve long-lived CAR-T cells. And so we're now capable of getting the necessary data points from months to years post-infusion and probe these persisting CAR-T cells if the product is actually able to achieve those or induce durable remissions in those patients in the first place. And so in a way, our study poses many more questions than we're actually able to answer. For example, like what is the significance of the double negative phenotype? Like why that phenotype in particular? The UPenn group had CD4 T cells, for example. What is the role of each of those genes in the signature? How can we adjust product manufacturing protocols to ensure every patient achieves or gets that signature in some way, which is almost like a biomarker of persistence? And part of that is looking for the top markers of that signature, like FXYD2, which is like a sodium potassium ATPase channel. How does that tie into T cell biology? And how does it tie into like CAR T cell long-term persistence? And so we can now put a spotlight on those genes that previously did not have any known immune functions, and we can start to explore what they're actually doing to support persistence. And so in the past, there's been lots of dogma around what long persisting CAR T cells look like, with the inference that these T cells might be stem-like or memory-like in their properties. But we can now see that their functional status after infusion didn't actually resemble that, but rather there's a strong basis in which to investigate the mechanisms of persistence using the newly defined gene modules that we've defined in this paper.
Well, Nathaniel, this is like amazing. And thank you so much for guiding us through your research and through your manuscript. And again, congratulations on that. It's a very nice achievement. And well, I think this is such an important piece of the biology of CAR-T cells, which is so much to be unraveled yet. So I really thank you. It was amazing to have you with us. And I hope maybe we can talk in another episode of the Trainee Pearls in the future, adding another piece to this puzzle.
Yeah, of course. Thank you. Again, thank you for the invitation.
Thank you. Bye-bye.
Bye.